Hello, everybody, and today I am here to start a new topic of biology that the chapter name is Cell, the Unit of Life. So, let's start with the cell, and here we are back with Mind Map series. Now, we will be covering all the four subjects, namely botany, zoology, chemistry, and physics. Hopefully, and now I'll be regular in my videos, and you should be regular in your support too. Now, let's start with the mind map. Come on. Firstly, let's move to the introduction that what makes an living organism that we are here to study in this lecture. The answer to this question says the presence of the basic unit of life, the cell, and all living organisms are composed of cells makes an living organism. Second question says cell is a fundamental and structural unit of life organisms. Anything less than a complete cell does not ensure in independent living. This is the answer to the question what is a cell? So, the first cell was described as a living cell by Anton von Leeuwenhoek. This is an important point, so you should remember this. Let's make a star mark on it. Let's moving further to the cell theory. In 1938, Matthias Sheldon, a German botanist, a botanist is a person who researches upon plants and gives their certain experiments or observations on plants, examined a large number of plants and observed that plants are composed of different kind of cells which form the tissue of the plant. At the same time, Schwann, a British zoologist, reported that animal cells had a thin layer called plasma membrane. This was first described by him that plants have a plasma membrane. So just mark this point as a star mark. And he concluded that plant cells have cell walls. Sheldon and Schwann together formulated the cell theory, but this theory did not explain as to how new cells are formed. Further, a new scientist named as Rudolf Virchow joined them and explained how new cells were formed. He said that new cells arise from the pre-existing cells or the cells that are formed preely or that are already existing in nature, and he termed this as omnis cellula e cellula, and finally modified the cell theory and gave its postulates as all living organisms are composed of cells and products of cells. All cell arises from pre-existing cells. This was the observation given by Rudolf Virchow, and this was given to us by Sheldon and Schwann. As you can see, he was a botanist and he was a zoologist. The year is also very short, and that he was born in 1838 and he was born in 1839. Don't worry, you will get the PDF of this with clear visualization and no highlighters marked. You just need to just put a comment down below PDF and I'll attach the link of the PDF. Now let's move further to over an overview of cell. As we know, egg of an ostrich is the largest isolated single cell. Nerve cells are some of the longest cells. See, what is the difference here? It says largest and it says longest. Largest can be whole diameter and longest means vertically it is larger or horizontally it is larger. It doesn't have a complete mass circulated over surface, but it is a cylinder form, vertical or horizontal shaped cell. Now we have further explanation of it as the cytoplasm is the main arena of cellular activities in both plants and animals. Ribosomes are non-membrane bound organelles found in both in eukaryote and prokaryotic cells. Apart from cytoplasm, they are also found in mitochondria, chloroplast, and rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome. Just mark these words with a different colored highlighter. Centrosome, which helps in cell division. This is its function. So you need to learn all these terms for a better visualization. A cell that is membrane bound nuclei are called eukaryotic cells that lack a membrane bound nucleus. If you want a full-fledged explanation video of this chapter, you can comment down below and I'll prepare that for you too. Till then, this mind map would be the best solution for this chapter's revision. Now further moving with the prokaryotic cells. Prokaryotic cells. These cells, like membrane bound organelles, are represented by bacteria, blue-green algae, mycoplasma, or PPLO. All prokaryotes have a cell wall surrounding the cell membrane except mycoplasma. This is the important point and can be asked in your question paper or your institute's test or your school test examinations also. So learn this point. In addition to genomic DNA, many bacteria can have small circular DNA outside the genomic DNA called plastids. Plasmid DNA confers certain unique phenotype characters to such bacteria. One such character is resistance to antibiotics. Plasmid DNA is used to monitor bacterial transformation with foreign DNA. The DNA is said in a common term such as double stranded circular DNA. Double stranded, that it is found in double strands. This is not DNA, bro. This is a centrosome. See, first strand, second strand, and it is found in SA this form. So it is double stranded and circular DNA. Now let's move further. Cell envelope and its modifications. Most prokaryotic cells have cell envelope which is tightly bound three-layered structure. Most prokaryotics have cell envelope which is tightly bound three-layered structure. Let me change the highlighter color as it is not visible. The most glycocalyx followed by the cell wall and the plasma membrane. The glycocalyx may be loose and sheath called slime layer or thick and tough called capsule. This should be on your tips and as it is very important to learn. The cell wall prevents bacteria from bursting or collapsing. The extension of plasma membrane into the cell is in the form of vesicle, tubules, and lamellae are mesosomes. It helps in cell wall formation, DNA replication, distribution of daughter cells, respiration secretion processes, and increase the surface area of plasma membrane. In cyanobacteria, chromatophores contain pigments. Each layer of cell envelope performs distinct functions. They act together as a protective unit. The plasma membrane is selectively permeable in nature and interact with the outside world. It is structurally similar to that of the eukaryotes. Bacteria may be motile or non-motile. If motile, they have flagella. Motile and non-motile means motile causes movement, non-motile doesn't cause movement. And for movement, they have certain structures called flagella, composed of three oats, filament, hook, and basal body. Villi and fimbriae do not play a role in motility. Bacteria, on the basis of the differences in the cell envelope, can be gram positive or gram negative. The bacteria who takes the stain on them are named as gram negative and those who do not take are named as gram positive. Further moving to ribosomes and inclusion bodies. Ribosomes are 70s, has subunits 50s and 30s. Several ribosomes have attached to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into proteins. The reserved material is stored in the form of inclusion bodies in prokaryotic cytoplasm. Example: phosphate granules, cyanophane granules, and glycogen granules. Gas vacuoles are found in blue-green algae and purple-green algae photosynthetic bacteria. If you are wondering here that ribosome is represented in 70s and 50s and 30s. So if we normally do addition of 50 plus 30 it gives out 80, but here it is the case of the when scientists measured it by putting the 30s ribosome here and 50s together, Rather of 80, it came out 70. So the ribosome is further named as 70S. Similarly, it will be the case for eukaryotic cells. Now, further moving to the eukaryotic cells. Besides the nucleus, eukaryotic cells have other membrane-bound structure called organelles like endoplasmic reticulum, Golgi complex, etc. The eukaryotes include all the protists, plants, animals, and fungi. Plant cells have large vacuole, animal cells have centrioles which are absent in plant cells. Ribosomes are of 80S in cytoplasm. Smaller unit is 40S and larger is 60S subunits. Cell membrane. Chemical studies on the cell membrane, especially in human RCB, enabled scientists to deduce the structure of plasma membrane. Cell membrane is mainly composed of proteins and lipids, mainly phospholipids. Phospholipids consist of polar head, outward and non-polar tail, hydrophobic inner side. In human RBCs, 52% of proteins and 40% lipids. Membrane proteins can be integral or peripheral, means they can be on periphery of the system or internally bound also. Let me erase this all. Yes. Most accepted model of structure of cell membrane is fluid mosaic model by Singer and Nicholson. This is the thing that you should remember. So I am placing it with different highlighter. Yes. Membrane is selectively permeable, means it allows only certain particles that it wants to take in. Many molecules can move across the membrane without any requirement of energy called passive transport. In this, no requirement of energy is required. Movement of water by diffusion is called osmosis. These are the important lines, so you should many molecules require energy ATP for their transport called active transport, example Na or potassium pump. Na is sodium and Ka positive is potassium ion pumps. The quasi fluid nature of lipid enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. Polar molecules cannot move through the non-polar lipid bilayer. The fluid nature of membrane is important for functions like cell growth, formation of intercellular junction secretion, endocytosis, cell division, etc. So, we have completed the prokaryotic, cells, eukaryotic cells in this cute and short video, and will be covering the next part of this mind map series of this same chapter. I'll be bringing all of these notes soon and will be making the video of this chapter soon. Till then, bye bye. Thank you.